This is part 3 and the last lecture for chapter 21. We were covering the supergroup Chrome alveolata. We had covered the alveolates because the Chrome alveolata are divided into alveolates and straminopiles. And for the alveolates, we'll go back, that included the dinoflagellates, one called Alexandrium causes red tides, the apicomplexans, and our example there is Plasmodium that causes malaria. Remember, their sexual reproduction part of their life cycle occurs in the mosquito and the asexual in the human. Ciliates included the one that causes fish ick and the paramecium. And then we went on to the straminopiles. The diatoms have a two-part glass shell. Golden brown algae. And then the Oomycetes, and here's a picture of a fish that is covered in water mold, basically. Okay. Then we have the supergroup Rosaria, which includes the cercozoans, foraminiferans, and radiolarians. They do all have pseudopods. This means the cercozoans actually can have pseudopods or flagella, but I'm probably not going to ask you about cercozoans. I tend to focus, and the questions in the quiz are on either foraminiferans or radiolarians. The foraminiferans have a skeleton or a shell known as a test, and their test is composed of calcium carbonate, which deposits of these foraminiferans formed the White Cliffs of Dover in England, I believe. But anyway, the pseudopods extend through openings in the test. Then radiolarians' test is also silicon or glass like the diatoms. And here's some pictures. So this is a foraminiferan, and then these are the White Cliffs of Dover. And then down at the bottom are the radiolarians. Okay, then we have supergroup Archaeoplastida, which includes the red algae and the green algae. The red algae are the multicellular seaweeds, although some of the brown algae is included in that. Mostly it's red algae that we think of as seaweed. For their economic importance, they are used to produce agar, which is used to make capsules for drugs, where like a powder goes in the capsule. Dental impressions, cosmetics, bacterial culture medium. We use it in the lab. We fill a petri dish with agarose gel and we grow bacteria on the surface of it. It's also used in electrophoresis to run DNA fingerprints. So it's like a gel. It's like a clear gelatin material. And it's used in food prep. Carrageenan is another one used in the production of chocolate and cosmetics. And then the reddish-black wrappings around sushi are porphyra is the name of the red algae. Here's a picture of red algae, and then there's a green algae. Most are unicellular, but some can be filamentous. Some are colonial. Chlamydomonas. I think it's unicellular. Yes. And Volvox is colonial, a colonial green algae. Here's some examples. Ulva is a multicellular green algae, commonly called sea lettuce. That's what it looks like. Charophytes are filamentous green algae, and the one that we look at in lab a lot, we have some slides of it. It's called Spirogyra, and here's what it looks like. Filamentous green algae. And that's more what this is what it would look like under our microscope. Okay, and then moving on to review, the supergroup Archaeoplastida includes the red algae, the green algae, and then it also includes plants, which are not protozoans. They're not in the kingdom Protista. They're in the kingdom Plantae. Supergroup Amoebozoa. Amoebozoans move by pseudopods. So the amoeboids feed by phagocytosis. They wrap their pseudopods around their food and bring it in, engulf it by phagocytosis. There is one called Amoeba proteus, and we do order those and bring them in and look at them under the microscope in lab. You've probably heard of the brain-eating amoeba, Naegleria fowleri, and then Entamoeba histolytica causes dysentery. All right, and here's just an illustration of an amoeba, and this is a pseudopod. Pseudopods change shape all the time. The amoeba is just constantly changing shape and moving. In fact, the only way to really understand this is to see it. So let's just watch one. This is only 30 seconds. I think you guys can see it. Yeah. Okay, so you see, this is called pseudopod, amoeboid movement, or this is pseudopods, movement by pseudopods. You can see the cytoplasm streaming along, and it'll stream along and change the shape of those pseudopods and form new ones. And the pseudopods also will wrap around food, engulf it, and then kind of form a vacuole around the food. And take it in and a lysosome will, and this is good. This is a amoeba that has been stained, but this is what it would look like if you were taking class with me. And we looked at it under the microscope in class. That's exactly what it looks like under our microscope, so that's good. Slime molds can be plasmodial or cellular. And I just want you to know that the slime molds are in the group Amoebozoans. Just make sure you know in the supergroup Amoebozoa that we have Amoebozoans, and then we have slime molds, plasmodial and cellular slime molds. Okay, then we have the Opisthocanta, which includes animals and fungi, as the two other kingdoms, and then the kingdom Protista, it only includes the choanoflagellates. The choanoflagellates are similar to what we call collar cells in sponges. So we'll come back to that when we get to the chapter on animal invertebrates. But this is what colonial choanoflagellates look like. They have like a flagella here, and they live in colonies, and the flagella helps move the water over this opening, and that brings food particles because they are heterotrophs.